Hello guys, welcome back to my channel. In previous video, I just described about it, what is the basic immunology and innate immunity. In this video, I am going to discuss about the adaptive immunity. What is adaptive immunity? Adaptive immunity is otherwise called acquired immunity. It consists of mechanisms that are stimulated by microbes and are also capable of recognizing non-microbial substance, that is toxins, called antigen. In adaptive immunity develops later after exposure to microbes and is even more powerful in fighting infection. In innate immunity, it is previously in our body, but in adaptive immunity, after exposure to the microbes only, the adaptive immunity develops. Types of adaptive immunity. First, humoral immunity and cellular or cell-mediated immunity. First, adaptive acquired humoral immune system. When an antigen presenting cells, APCs, that means antigen presenting cells are mostly macrophage and dendrite cells, present an antigen to B cells. The B cell is signaled to proliferate and produce the antibodies that specifically bind to the antigen. Okay, antigen presenting cells present on antigen to B cells. The B cells give a signal to proliferate, produce antibiotics. If antibodies bind to antigen, it acts as a signal for polymorphonuclears, that means leukocyte macrophage, to engulf and kill them. Coming to adaptive acquired cellular or cell mediated immune system, that is involved in T lymphocytes. The T lymphocytes are specifically made in lymph nodes or responsible for acquired cellular immunity. If the antigen presenting cells present the antigen to T cell, the T cells becomes activated. Activated T cells proliferate to produce CD4 T cells and CD8 T cells and killer T cells. Coming to humoral immunity, that means B cell development and formation of antibodies. The B lymphocyte is responsible for forming antibodies, we already seen, that provide acquired humoral immunity. In early development of B lymphocytes occurs in bone marrow. In the first step, stem cells develop into pro-B cell, and in response to signal interleukin 7 received from the stromal cells in the bone marrow, it promote differentiated into pro-B cells and pre-B cells. Okay. In upcoming slides, I am going to show the slides. The photos, then only you can clearly understand. Okay, wait for it. And the pre-B cell undergo proliferation. This give rise to B cells with monomeric IgM, IgD molecules. That is called monomeric immunoglobulins. It is otherwise called surface immunoglobulins. The surface immunoglobulins IgM and IgD present on the surface of all native B cells. It constitutes the antigen binding component of B cell receptor. B cell recognizes the antigen via B cell antigen receptor. In addition to membrane immunoglobulin, the B cell also express several other molecules that are all complement receptor, Fc receptor, and then CD40. When virgin B cells reach secondary lymphoid tissue, they may be activated by protein and non-protein antigen. The activation of B cells require help from helper T cell. Helper T cell activate B cell by binding CD40 expressed on B cells by secreting cytokines. This interaction is essential for B cell maturation. Different cytokines stimulate B cells to produce different antibody producing cells. The end result, B cell activation, is the differentiation into antibody secreting cells called plasma cells. The plasma cells then only produce the antibodies. Here I am showing the picture of it. First, bone marrow is the hemopoietic organ. So bone marrow produce the stem cell. They differentiated into lymphoid stem cell. Again it further differentiated into pro-B cell and pro-T cell. The pro-T cells migrate into the pro-T cell into the thymus. Here the pro-T cells migrate into the pro-T cell into the thymus. In the thymus, the further maturation leads to it form helper T cell and cytotoxic T cells and suppressor T cells. In the bone marrow, the pro-B cell further differentiated and proliferation with interleukin 7, it form pre-P cell. The further proliferation, it form the monomeric immature B cell with monomeric IgM and IgD molecules on the surface with the help of helper T cell. See, you can clearly see here. The further maturation, it form IgM, IgD, IgE, IgA antibody producing mature B cells. And coming to the B cell stimulation and how specific antibody secreted. The B cell is triggered when the antigen bind to B cell. The B cell engulfs the antigen and endocytosis and digest it from fragments of peptides. In upcoming slides, I am going to show the picture. Then only you can clearly understand. Okay. This degrade pieces of antigen bound to unique class to major histocompatibility complex molecule displayed on the cell surface and with the formation of antibody producing activated B cells. In human, major histocompatibility complex is called human leukocyte associated antigens. Okay. Here you can clearly understand the activation of native B cells for production of plasma cell. How the plasma cell produce antibodies. First, this is the mature B cell which contains immunoglobulin of the B cell receptor complex. The surface contains a surface immunoglobulin of B cell receptor complex. When an antigen binds with the surface immunoglobulin, it engulfs the complex. So here you can see the endocytosis of the receptor antigen complex, and in this surface immunoglobulin B cell complex, it fragments the antigen into peptides and synthesis the complex of class 2 major histocompatibility complex glycoprotein. Here you can see here the green color is class 2 MHC glycoprotein. The purple is antigen fragment. This fragments of antigen become displaced on the surface bound in the groove of the M class 2 MHC molecule. It forms processed antigen displaced on MH2 molecule. So it is the activated B cell for formation of antibody producing plasma cells. The activated B cells then binds to helper T cells which possess CD4 protein. When this happens, cytokines are secreted by the helper T cell and helper B cells to multiply and mature into antibody producing plasma cells. The plasma cells then secrete soluble antibody corresponding to the surface immunoglobulin found on stimulated parent cells. Here you can clearly see this is the activated B cell with the MHC2 complex and it binds with helper T cells. Here you can see the B cells and helper T cells It sends a signal to secrete the cytokine to stimulate cell multiplication and maturation of the antibody forming plasma cells. These plasma cells further produce antibodies released into the blood. Each antibody is specific for particular antigen because it's a unique structural organization of amino acid in the variable portions of both light and heavy chains. There are two variable sites on illustrated antibody for attachment of antigen and making antibody bivalent IgM, which consists of 10 light chains, 10 heavy chains as many as 10 binding sites. For IgM it is the bivalent, for IgG it is the pentavalent. Here you can see this is the antigen, this is the antibody IgM, it is the bivalent. Antibody of antigen molecule to one another by bivalent antibody. Coming to the mechanism of action of antibodies. Antibodies act mainly on two ways: direct action, indirect action. Direct action by direct attacking of antigen. By indirect action means activation of complement system. Here, in direct action, the bivalent nature of the antibodies can inactivate the antigen agent in one of several ways by binding the antigen via antigen binding region of antibody. This can lead to any of the following methods: by agglutination method, by precipitation method, by neutralization method, and by lysis. In agglutination method, it is in multiple large particles with antigen on their surface, such as a bacteria or red cells, bound by cross-linking together into the clump. In the precipitation, molecular complex of the soluble antigen and antibody becomes so large that is tender to insoluble and precipitate. Okay. In neutralization, by blocking of attachment of virus or bacterial toxin to the membrane receptor by covering the toxin site of the antigenic agent by antibodies. In lysis, in which some potent antibodies are occasionally capable of directly attack the membrane of the cellular agent and thereby cause rupture of the antigen. In indirect action, most of protection comes through the only indirect action by activating the complement system. The activation of the complement system is initiated by binding of one of the complement molecule of Fc portion of the antibody molecules. That's it. And coming to development of T lymphocytes, why we are going to see now T lymphocytes mean adaptive cell mediated immunity. Okay, the T lymphocytes are derived from stem cells of the embryo. I already showed. Okay, the stem cells of the embryo T lymphocytes. The stem cells themselves incapable of forming directly activated T lymphocytes. They are further differentiated into thymus. Lymphoid stem cells after formation in the bone marrow or after first transported to the thymus gland. I already showed first bone marrow contains a stem cell and the stem cell further differentiated into the pro P cell and pro T cell. In the pro T cell migrated into the thymus to form the further T lymphocytes. In the thymus gland, they divided rapidly and at the same time develop extreme diversity of reacting agent against different specific antigen. The different types of proposed T lymphocytes then leave the thymus and are distributed via blood throughout the body and stored in the secondary lymphoid tissue everywhere. Millions of specific T lymphocytes stored in the lymphatic tissue. When specific antigen come in contact with T lymphocytes in the lymphatic tissue, the specific T lymphocytes become activated and form mature T cells. The T cells are then distributed throughout the body. It is the killer T cells, CD4 cells and CD8 cells. The major three types of the T cells are CD4 T cells, that is helper cells, and CD8 plus T cells, that is called cytotoxic cells, and killer T cells. Now I am going to discuss about the role of helper T cells. What is the role of helper T cells? As their name implies, it is the help in the function of the immune system by several ways. The activation of the helper T cells causes it to release number of protein called lymphokinase and cytokinase, or more specifically interleukins. The helper T cells only release the lymphokinase, cytokinase and interleukins and other stimulatory signal to stimulate the activity of macrophage, killer T cells and B cells. Helper T cells express TCR. TCR means T cell receptor that recognize antigen bound to class 2 MHC molecule. I already shown in the antibodies formation. Helper T cells have no cytotoxic activity and don't kill infected cells or clear pathogen directly. It just only help to produce the signals. They instead control the immune response through lymphokinase by directing other cells to perform this task. Lymphokinase act as a regulator of immune function by acting on other cells of the immune system and bone marrow and help determine which type of immune response the body will make to particular pathogen. In the absence of them, lymphokinase, the immune system is almost paralyzed because the helper T cells only produce the signals like lymphokinase and cytokinase. It is further acting on B cells and T cells. Then only it produces the antibodies. If there is any absence of the lymphokinase, the almost immune system is paralyzed. Here you can clearly see. First, antigen act on helper T cells. It produces feedback stimulation, further release of helper T cells, and it activates the macrophage by the producing signals like interleukins, and it all stimulate the cytotoxic cells, and it stimulates the suppressor B cells, and also it stimulates the proliferation and the differentiation of the B cells by the producing the signals of interleukin cytokinase. It leads to formation of the B cells. The B cells only produce the antibodies like IgM, IgG, IgA and IgE. And coming to the role of cytotoxic T cells, killer cells and CD8 T cells. Cytotoxic T cells is called killer cells because they are capable of killing directly certain tumor cells, viral infected cells, sometimes the parasite also. Cytotoxic T cells directly attack cells carrying certain foreign or abnormal molecule on their surface. Here, cytotoxic T cells recognize small fragments of the viral proteins carried on the surface of infected cells by body own class 1 IMC protein molecules. The protein of the infecting virus processed into fragments which combine with class 1 MHC protein molecule and brought to surface of infected cells. These are complexes recognized by the killer cells. In picture I am showing to you, you can clearly understand. Okay. The killer T cells are activated when the T cell receptor bind to specific antigen in a complex with the class 1 MHC molecule of the another cells. After binding, the killer T cells secrete, forming perforins. The perforins make a hole in plasma membrane of the infected cells, so it leads to interstitial spaces filled with fluid, and it becomes swollen and dissolves shortly thereafter. Killer T cell activation is tightly controlled and requires activation of signal provided by helper T cells. Here you can clearly see direct attack on invading cell by cytotoxic T cells. This is the infected cells by the virus. In the cells, the viral protein is synthesized. The viral protein is processed into pieces. The viral protein, here you can see the viral protein fragments. First, the viral protein is synthesized and it forms the viral protein fragments. The fragments are attached with MHC1 complex, and also the killer cell contains T cell receptor. It is attached with MHC1 complex with CD8 cells. So it forms the cell perforins and leads to invert of the fluid into the infected cell, and the infected cell is swollen and rupture of the cells leads to death. And coming to the role of the suppressor cells. As their name implies, they are capable of suppressing the function of both cytotoxic T cells and helper T cells and prevent excessive immune reaction that may cause damage to the own body tissue. Okay, the role of suppressor cells means it suppresses the both cytotoxic T cell and helper T cell to prevent excessive immune reaction. Thanks for watching. In next video, I am going to discuss about the vaccine and immunology memory. Subscribe for more.